spilled over and it was unavoidable. But it's great to be part of this very, very exciting initiative. And I'm going to talk today about some principles that have this big heading of an algorithmic approach to biological structure formation. And I think the two previous talks have already set it up very nicely for me to proceed over here. First of all, just to get everybody excited, here are two people who have very, very broad views of science, Freeman Dyson and Paul Nurse. These quotes, one from 20 years ago and the other one more recently, sort of indicate the fact that biology has not shied away from interdisciplinary ideas. I mean, it's had a very long history over hundreds of years. And Dyson says now, in the 20th century, in 21st century, a lot of people who are really computer scientists are doing biology. The line between mathematics and biology is not so sharp anymore. And Paul Nurse alludes to this idea that you should look at biology in terms of processes and mechanisms rather than as something static. And I think these two ideas have really been quite central to the way we've approached biological questions. Very, very broadly, for me, biology looks like this. So in the middle of the triangle, you have physics and chemistry, and those things provide the constraints. Those are the laws that are immutable, and you have to deal with how molecules work and second law of thermodynamics and so on. But the things on the outside, those are the principles: information, evolution, and I put cells over there. It's a little controversial whether all of life is made of cells, but I think these kinds of very broad principles combined with the constraints of physics and chemistry really allow you to think about biology very broadly. I think a lot of the questions that came up after the previous two talks are about how you jump between different scales. And the fact is that information at one scale really does propagate both up and down in biology in very, very non-trivial ways. There's no dearth of open problems. So here's a sperm whale, and we don't really know how it's put together. But we don't also completely understand how proteins are put together. And so there's really a lot of work to do. And I should say, just because we know how proteins work doesn't mean we understand biology. What's very interesting to me about this whale example, of course, is the whale is made of the same fundamental molecular building blocks as we are, but it's so different at an obvious macroscopic scale, but it's also different at all scales going down. For example, whales have a version of hemoglobin, which you and I have, which have atomic level modifications so that it's able to work with the kind of oxygen saturations that are available at depth. Okay, so changes happen at all scales in all organisms. And you really need to piece together that entire chain of causality to understand what the constraints are and how things work. So this is my big, to get people excited. So structure formation, whether you want to form a whale or form a protein, there are many, many examples in biology where structures are formed. And the idea is that you want to think about that as a process. Obviously, it's not a given structure, but it's assembled from smaller components in every case, right? And there are many ways to imagine making a structure. For example, there could be very specialized machinery that's designed to make a single specific product from a single input. Okay, it's very narrow and it's only able to do this one job, and that's not the kind of thing that biology is really built on. Biology is really built on things that are adaptable across different contexts. Simple example of that is from RNA, messenger RNA, you can make different kinds of proteins because the RNA can have different sequences, and there's a machine called the ribosome which translates those RNA sequences into protein sequences. But that's also sort of not the most complex thing you can imagine, because it's really templated, right? It's like having a mold and making a sculpture inside that mold. If you have some version of it, you can easily make sort of a copy of it. DNA replication works in the same way. Most of biological structure formation actually happens in a more complex way. It happens because there is, I'm going to call an algorithm, and I'll describe what I mean by that in the next slide. But basically, it's a process. You take building blocks, you put them in. There's some complex time dependence and spatial dependence. All kinds of things change environmentally and at microscopic levels, and you produce some final product. We've been doing work about how that kind of algorithmic structure formation happens at the molecular level, but it's really a metaphor for how a genome creates an adult. The adult is not a copy of the genome. There's no homunculus, right? The adult is really made by a recipe that's somehow embodied in the genome in ways that we're only beginning to understand. So for me, what's an algorithm? I'll describe it by example. So in fact, in the last 10 years or so, there's been a lot of progress in using to build higher order structures. You have self-replicating clusters. You can have what I call multifarious mixtures, where you retrieve structures of various types from a single nucleus. And you can actually self-assemble, rather things that reach the meso macroscopic scale from these basic molecular interactions. How do they do it? Of course, it's a process, right? It's not templated, but it's not any old process. It's an algorithmic process. An algorithm for me is a constraint on a process where if you start from some input state, there are many ways to go because things are stochastic. And what you really do is you cut off paths that take you away from where you want to go. You encourage paths that take you where you do want to go, and then you cut off paths that take you forward from there. So you reach a target and then you don't go further. I know I'm talking very abstractly, but for example, suppose you wanted to build the Eiffel Tower just based on molecular interactions. What molecules would you make, and how would you remove these off-target paths in order to get that thing to work? And in this field, there's huge amount of open questions. There are inverse problems, what materials you program. There are simple forward problems, given these building blocks, what do they make? I'm going to give you just two very quick examples from our own work before I finish the talk. So here's some very beautiful EMs of a eukaryotic cell. Those are the kinds of cells you and I made out of. And the structure you're looking at is called the Golgi apparatus, or the Golgi body. It's really pretty complicated, and it's a snapshot of this was in my title slide. So we're interested in the Golgi, not only in what it does, but how it gets made. So what is the Golgi? If I were to abstract it out very much, this nice review covers it. The Golgi is a series of compartments within which there are enzymes that build complex molecules. And these series of compartments function like a factory production line. These molecules are handed over from one compartment to the next, to the next, to the next, in order to build a complex molecule. So the cell has sort of created the structure in order to control the assembly of molecules and cut off-target pathways. One of the things we showed a few years ago was that the creation of this cellular structure actually emerges from various prescriptions at the microscopic scale in terms of molecular interactions. I won't explain what's on the slide, but basically these graphs that I've shown here with these nodes and edges are sort of an abstraction of a cell. Each gray blob is a place in a cell, is an organelle, and the arrows show how things move around between them. And biologists have taken a lot of trouble to understand what the map of a real cell looks like. Now, it turns out that by encoding the right molecular interactions, running the system, this is a simulation, but you could also do it in vitro, you actually get structures of sufficient complexity that resemble real cells, right? So this is a sort of proof of concept that a real cell is encoded by a process at some structural level. The other work we've done more recently is shown that once you have this compartmentalized milieu, you can actually use the idea of this factory production line to create very complex molecular assemblies, these little yellow circles and blue squares and so on. These are called glycans, and they're very, very important in how cellular biology works. It's very hard to imagine how to make these complex structures to order of the right shape and size if you have just one pot, like the chemists talk about one pot. But if you have many pots and you're able to control things from one pot to the other and control the transition time, you can actually build things correctly. And again, there are inverse problems here that we can solve, and we've shown that taking an algorithmic approach is really the right way to go. So I'm going to finish with a high-level thought that none of this actually works in abstraction. I think the right kind of place to do this kind of thinking is when you're embedded in an experimental biology environment. And I think NCBS, the reason I love it is because it really allows this sort of interdisciplinary interaction between multiple groups working at multiple scales, and allows a theorist like me to jump up and down between all those scales. So I'll end over here. The little cartoon on top is a picture we commissioned a few years ago for the Simon Center where I work. On the left side is this logo turtle, if you remember the logo programming language. And on the right side is a real turtle, or at least a picture of a real turtle. And I think this multi-scale idea captures what we're interested in. Thank you.
Hi, Mukund. Sorry, Shravan here. Can you explain a little bit about how you think machine learning or computer science
So as you see, I didn't put machine learning in there, and it was deliberate because we haven't actually used it. But what I think its role is not so much in the sort of application-oriented framework, but in really understanding how processes like this. So it's really solving the inverse problem. Okay. So what you have are a huge number of ways in which to design building blocks at molecular scales and design their affinities and sort of design time-dependent environmental conditions for them to grow. I mean, the space is enormous. And what you want in this case is to build structures of some given size and shape and maybe physical properties. And so far, the way that's been done is by heuristics. Okay, so people have attacked very simple structures, and they found certain principles in terms of the number, the size, the shape, some temperature, those kinds of very simple external parameters that can be used to shepherd these processes out to a final conclusion. But of course, in a cell, it really doesn't happen like that. Things are much more fast and noisy and out of control, and yet the whole structure gets built, right? So I think what we could do is, and since I'm not an expert in ML, this is all just speculation, we would like to encode these problems in ways that machines understand, show them a few examples of how it's been done, and then extrapolate to larger systems and structures and ask what kinds of building blocks would you need to achieve certain outcomes. Okay, I'm sorry I can't be more specific because I'm just getting into this, but that's the rough idea.
So, hi Mukund. Just because you have framed some of your description in the form of paths being taken and a target being achieved in some way at some point in the path, like, is there any work already using reinforcement learning in this domain, or at least problem formulation using RL?
Using reinforcement learning. Okay, I see. So training by comparing to targets and
actually some trajectories being tried out.
Right. I'm not familiar, but I'll tell you that the work in ML that's been used so far is not so much these kinds of bespoke single structures, but for example, crystal lattices with multiple components that have certain properties. People have used machine learning to identify a prescription to make lattices of given shape and size, right? And that's also, I think, is being tested experimentally. I have to understand more clearly how reinforcement learning will work here. We don't yet have such a clear problem statement, okay, that we can jump right into a machine. And so what, in fact, Shravan and I and Srikanth and others have been trying to work on is narrow down the problem from this very abstract picture that I put into something that is more tractable and something that is more amenable to a simple machine learning approach.
Thank you.
Okay, thanks a lot, Mukund. I think we'll go to the next speaker. Thank you for your talk.
Thank you.
Yes, ready to proceed. Yeah. Thanks for the invitation. So let me start by saying a few words about my background. So my work is mostly in probability and computer science, and often on connections between phase transitions in physics and algorithms, and sometimes touching on information theory, and also on probabilistic graphical models and causality. The closest I've ever come to all of this was a euphemism to say that I'm not a biologist who is in the unenviable position or enviable position, I don't know, of giving a talk about biological problems. The closest I've come to biology before this is doing some mathematical models of evolution. So, but let me start nonetheless. So the problem I want to look at is about the structure and dynamics of certain biomolecules. In particular RNA, and this is a project that we have recently started with Sachi, who's at NCBS, and Hari, who's also in TIFR Mumbai. So So the problem here is sort of well known. It has been mentioned already a few times in the talks before this. And it's about going from a sequence to a 3D structure. That's usually the more famous version of this is for proteins. So you have amino acid sequences and you want to go to the 3D structure of the protein. The version we want to look at is where you have nucleotide sequences, just 4 letters in the alphabet rather than the 20 odd for proteins. And you want to go to a 3D structure. The questions that you want are similar as the protein case. So you want to find what the final structure is, the stable, somewhat stable structure that comes from a sequence. And the second one is this, which I'll come to later, which is you want to also know how this stable structure is actually achieved. So somehow there is a molecule, there's a big molecule. It starts off in some configuration, but it slowly comes into this final structure. The question is, how does it come to that? It doesn't sort of start off there. This will come to later. So here is just an example of what such a 3D structure might look like. So this is for a very small RNA fragment. It's actually from 2001, the structure. So it's you can see it looks a little bit complicated, but maybe now it will look a bit simpler. So this is actually the RNA chain. This is what corresponds to the sequence, and the other things sort of attach onto it. So we can talk about more detail about this. So why should one do this? So we've all heard about AlphaFold already multiple times, and the thing here is a more recent result which I found quite interesting. So here, so let's forget about the machine learning thing. So what they did really is this. So they had a neural network. The neural network existed 2 years before that. They had a neural network which takes not the sequence, but actually the structure of a protein, of a small part of the protein as input. And it tries to do somewhat like an inverse problem. It tries to see which are the viable amino acids that can inhabit that local environment. And that lets them know what are the viable mutations they can make to a particular structure. So that's just one part of the thing. Then using this, so they could whittle down to which are the viable mutations and get a mutation which does much better at eating plastic. So it's an enzyme that eats plastic, but it was achieved through finding these viable mutations, which is from a sort of a neural network that is solving the inverse problem of this, going from structure to one location and saying, what can you change this? Okay, so and this neural network it's not, as things go, it's not that complicated. It's big. There's 4 so-called 3D convolutional layers, 3 fully connected, but it's nowhere like GPT or. So for RNA, it's different from proteins, and there are many reasons that make this problem different. So one is that there's much less data. So this is just a numeric comparison. This is actually the last time I checked 2 days ago, about 190,000. That's like 2 lakh protein structures, and just 6,000 odd for RNA. And the other thing is that for the thing you want to do, making this backbone structure, RNA structure is far less constrained. So proteins, because of the nature of the covalent bond involved, there are many more constraints on what the backbone can do. Famously, the Ramachandran plot captures some of this. There's far fewer constraints in the RNA world, and so the number of sort of local angles you need to keep track of is much larger. So here is what has been done for RNA. So here's another recent paper. So it again. It takes a sequence, puts it through some neural network, and gets these torsion angles per site. But let's see what it does. So they don't say how many parameters they have. I tried to put it into TensorFlow given what they were describing. It's about 30 million. Again, not that big by these standards. But the funny thing is that it's trained on about 300 sequence of median length 80. So it's not a lot of data. So the training time was probably not that high because there's not too much. That's not enough. There's so much data to run on. They get decent results based on just this. The network was not too complicated. I didn't mention what it is here, but it's inspired from these language models. But it's less complicated than the mute compute. Okay, so before I go further about what we want to do, I think this has come up in this thing. So one important concern I have, at least whenever I see machine learning being applied to science, is that is it a case of this, that there's a hammer and you want to put, and you have a nail and you want to put it. But I would like to say that maybe it's slightly different. It's like, it's somewhat more principled. There are many hammers and there is a particular nail. and you want to see which one is the best one, and you want to do that both theoretically and experimentally. You should have some a priori reason for trying a method. So, for example, the previous ones that they were using language-based models, but there is some rhyme to it in the sense that those language-based model try to capture. So they try to capture sort of correlations across several length scales, which is true, which happens to give language its complexity, but it's reasonable to suggest that that also gives these big molecules complexity. So as I said, I'll talk about coarse graining here. Again, coarse graining is something that was mentioned before, but what is it useful for in this context? So this is again the structure I showed you earlier. So this is the backbone. But there's so many of these other atoms hanging around. So if you wanted to do this other problem of trying to see how you get to the structure, and you want to and you started doing an all-atom simulation on this, that will be too slow. So what is often done is that you focus on a few atoms, and these might not even be real atoms. These might just be some center of mass type objects, and you only take those and you try to simulate just with those and with some potential functions that are geared to just see how these virtual atoms evolve. And then that it loses information, but it can be faster. And so one thing we would want to do is to use the structure prediction. So this is what we want to do. So we have not done anything so far. So that's another thing I should have said. So it's more about what we want to do now and what we've been exploring. So there is structure prediction, there's better coarse grain simulation, but they're related problems in the sense that the predictive structure, these methods for predicting structures can help you whittle down what are the correct coarse grainings in some ways. And they can also let you figure out what should be the starting configuration in some sense, where you should start. I mean, so these structure predictions are approximate, so you could try to even make them better by running a simulation from them. So that was all I was going to say about here. And are there any questions? If there are no questions, I'll ask a very embarrassing question myself. So so that's okay.
Out the representation, like what should be coarse grain based on some machine learning, or are you just using the structure to get?
So right now, so the one thing we would want to just do is to go between coarse and fine grained using learning techniques. So the learning technique does not come into figuring out what the coarse graining is. I mean, it helps it in the sense that if you know the structure predictor, it tells you what the angle should be and so on. But after that, the main problem would be to go between coarse and fine grained structures.
And that's currently not a learning object.
So yeah, so people have done things which will look like learning, like clustering and so on, but not with networks.
So RNA structure is dependent on basically size of the RNA as well. So are you trying to look at diverse sizes?
Yes. So as I said, like there are only about 6,000 sequences. Correct, yeah. And so for example, in this one, the result that I mentioned here, this one. So I said that their median length is about 80, and their highest length sequence was about 400 something. So that. And the reason, I think, if I recall correctly, the reason the larger ones were discarded is because parts of them were very, very similar to the things they already had. So it would just bias the learning. So these kind of problems are also there about because the data is so less that you cannot do many other things.
Well, I'm going to show you in the smaller data set later.
Sorry?
Smaller data set than yours. I'm going to show you in smaller data.
Oh, I see. Okay. So maybe I should ask. Sorry. Yeah. Okay.
Okay, can you all hear me? Yeah. Okay, so I'll start with a disclaimer. I'm an experimental biologist. I know next to nothing about AI other than that I think it will be useful for the kinds of things we do. So I would really like to tell you a little bit about what sorts of things we do and what we think AI will help us with. So broadly, we're interested in the problem of morphogenesis, and just some definitions, given that this is a sort of diverse audience. Morphogenesis is essentially the generation of form, and morphodynamics is refers to the changes in form as a function of time. That better? Okay. And mechanics refers to physical parameters, including stresses, strains, and stiffness, and molecules, I guess, is sort of self-explanatory. And we look at morphogenesis in the context of the development of an animal, and in this case, the maggot of the fruit fly. And as you will see in the schematic on the left-hand side, the life of a fruit fly starts as an ellipsoid egg that is sort of a hollow structure. Okay, that's a hollow structure covered by a single sheet of cells, these gray dots are the nuclei here. And pretty much everything that happens to the egg till it sort of becomes a larva, which you will have seen inside fruits and so on, is associated with the deformation of this single sheet of cells by forming folds, by forming bends, furrows, and so on and so forth. And we're essentially interested in understanding how such changes in form that are dynamic over the course of the development of an organism are powered and how they're choreographed. And one way in which we study this is to parse this entire process into steps that we then study in detail. And here are two processes that we've tried to focus on. And I just wanted to highlight here that if you are wondering why the fruit fly is worth studying at all, I'd just like to say what I think Google said, which is that DeepMind is to AI what Drosophila is to biology, and that's sort of what has kept us interested in looking at how the fly does it, because a lot of how every other animal does it is similar. So what recent advances in microscopy have helped is to understand or visualize this process of morphogenesis in real time. And here you'll see okay, there's some delay here. Okay. You'll see every single nucleus on the surface of this animal labeled here. And you'll see that as the animal develops, you'll see that groups of cells are, clustering together, or sheets of cells are moving, in this case, backwards, or in this case, towards the center. And you'll see that there's a lot of heterogeneity, not just in the way cells behave, but also in the way groups of cells deform and reposition themselves. And so what we've been looking at is how these morphogenetic changes are brought about. And in particular, we've been interested in asking what the interplay is between molecules and forces, and how you integrate information at multiple scales, from molecules and forces to cells to cohorts of cell neighborhoods to eventually sculpt tissues. And I just want to say that the sheets of cells we're thinking about are actually mechanically connected cells that look something like this if they're very ordered. But in fact, these, as I told you, the embryo is ellipsoid, and so this sheet of cells is actually wrapped over a curved surface. So the cells actually don't really look like this, but look somewhat like that. And the way the cells sort of change their shape and move relies on the activities of intracellular proteins, notably polymers of actin and microtubules in conjunction with motor proteins. And these forces that they generate are transduced to the outer surface of the cells, the cell membranes, through their links to proteins on the plasma membrane here. And so this is broadly how cells can deform, and we're interested in asking how cellular deformation is used to sculpt tissues into different shapes, and how molecules and forces contribute to this. And so what is the sort of data we generate in the lab? So our work is either exploratory. We ask, okay, let's understand how this tissue moves or becomes repositioned during the course of morphogenesis, and we begin by visualizing it in an exploratory way. Or we might have hypotheses and that we then try and prove or disprove. The samples that we have are animals that are going through this process in an unperturbed way, or we introduce perturbations to it that could be of a genetic nature or a mechanical nature. And our sample size, because of the time scales involved in imaging, as I will show you, is not big or as big data would demand, but our sample sizes vary between several tens to hundreds of animals. But if we're looking at cells within tissues in an animal, we're looking at several hundred to 1,000 cells. And the sort of data we generate, of which I'll give you samples of, are high temporal spatial resolution, real-time 3D image data sets that we often view as 2D projections or as orthogonal sections to get 3D information. They're often multiply labeled, so we can look at many things at the same time. For example, the membrane and the cytoskeleton. And we can capture dynamics at multiple length and time scales. We can quantitatively analyze the dynamics of morphology, cell position, where the labels are located, and what the intensity of these labels are. So I think the data we have, although not big, is pretty rich and has high order spatial and temporal information with regards to shapes, patterns, textures, locations, and neighborhoods. And we've used this sort of data effectively with theoretical physicists, originally Madan Rao at NCBS, but more recently Vijay Krishnamurthy here and Mandar Namdar at IIT Bombay, to extract physical parameters that include order, packing, coherence of groups of cells, stresses, strains, and flows. And so what I think I'd like to say is that the data we have is rich, but I think we would like to be able to better extract the rich information encoded in these images and be able to integrate data not just from multiple samples, but also from multiple scales within the same samples. We would like to be able to discover correlated patterns, for example, patterns of cell dynamics and cytoskeletal dynamics, and then sort of use our usual approaches to test their biological relevance. So what I thought I would do is sort of give you a sample of the kind of data we generate. So here's one part of that whole scheme of morphogenesis, which sort of results in the closure of the embryo. So you'll see an elliptical sheet here made of polygonal cells that contracts to bring the cell sheets that it's attached to close to each other, where they end up meeting in the middle and fuse, right? So this is imaging. This is 3D visualization. This is a 2D projection of the 3D data set. And we've several biological questions we're interested in understanding. But what I want to show you, if you zoom in on that elliptical region, is the sheer complexity of not just the static shapes, but also the dynamics of the shapes. And you will see that these shapes can't be described as just polygons, because over time they sort of accumulate these squiggles on their membranes and so on and so forth. So one of the things we really like to be able to do is to classify complex cell shapes and cellular interface dynamics. So you will see that the areas of contact between two cells can increase or decrease. What we would also like to do is to sort of be able to classify this heterogeneity in shapes in an unbiased manner, so we can work out whether these are part of a continuum or they're discrete shapes. We would also like to be able to predict some behaviors that appear stochastic to us, and understand whether the asynchrony in the dynamics of these cells actually, for example, are asynchronous, or there's some hidden pattern of communication or propagation between cells. So I think what we would like, therefore, to use AI to understand is this interrelationship between the shape dynamics of adjacent cells, and try and understand how the neighborhood influences it. So far, we've been able to do a lot of things quantitatively, but not in sort of high throughput. We can analyze the areas of cells as a function of time and define phases and pulses. We can correlate the pulsatility of the cells with the pulsatility of the cytoskeleton and signaling molecules. We can visualize with fairly high resolution the patterns of behavior of these cytoskeletal elements, but we can't. I mean, we would like to be able to use AI to uncover hidden patterns, for example, asters and changes in orientation that I think we cannot actually do a good job of. So I think what I'd probably just like to end by saying is that we would like to be able to extract data from the images we have, which we believe are very rich. We would like to do an unbiased classification of shape and dynamics to identify discrete clusters or continua. We'd like to discover new correlated patterns, test their relevance, and if we set up a pipeline for extraction, integration, analysis, we can use it for other things we study, and each movement we study has a parallel in humans, and we believe that such an analysis can help us understand how our body axis is straight, how our organs are positioned, and why some of these disorders, like open neural tubes and cleft lips happen, and why sometimes wounds don't heal very well or cancer spread. So that's broadly all I have to say.
Good morning, everyone. Title of my talk is protein folding and design and for making mechanically robust protein-based materials. So when I saw the poster, basically it's given importance to proteins because its AlphaFold is actually became very well known in the last couple of years because of the protein folding. Why proteins are actually important? Proteins are beautiful and essential molecules in living systems, and they are important for multiple functions, like in neuroscience, like UP mentioned, or Shruti mentioned in other cases, and also sometimes even catalyzing reactions other than RNA. They come actually in multiple varieties. And what are these proteins? These are nothing but polymers of amino acids, and these amino acids have actually different flavors. So they come in charged, uncharged, polar, hydrophobic, hydrogen bonding, etc., etc. But what's actually important state of these proteins is actually the folded state, where these biopolymers, which don't have any structure to begin with and fold into a well-defined state in most of the cases. In some cases, they are like IDPs, like Shruti mentioned. But in most of the cases, they actually fold into well-defined structure called native state, and this is functional. So if these proteins don't really fold into these states, and then sometimes they misfold and then they aggregate, and then they create actually problems. So in some cases, these aggregates can be cleared because of other proteins inside the cells, or sometimes these proteins actually aggregates and they can't be cleared, and then they create a disease state, some like in Alzheimer's and Parkinson, etc. So AlphaFold is actually became well known for predicting structures from the purely based on the amino acid sequence, because there are a lot of structures available in the PDB. So AlphaFold actually learns the structures of all those existing proteins, and then if you submit a sequence, then it tries to find out the structure and actually does very good job. But what is not really known is actually how this unfolded chain actually goes into folded state. What are the intermediate steps? Okay, what are the sequential pathways in which these proteins actually get into this structure is still not known. It's still unknown, unsolved problem called protein folding. So today I'm going to talk about one specific function of the proteins among many different functions. Is basically function which is associated with stretching force or mechanical force. So if you see these two pictures here, so the one is both are taken from the natural world. So one is basically muscle tissue in us, and another is basically spider silk. So at the prima facie, they look actually completely different from each other, but there are actually some commonalities between these two systems. Is that both are actually mainly made up of protein molecules, and then these are actually have ultimate strength. So if you see the spider silk, if you see these strands, they have actually thinner than human hair, roughly 3 orders of magnitude smaller or 1,000 times smaller, but they are actually so strong that they're actually stronger than steel, okay, for the same dimensions of steel if you take. But how actually these molecules or this material made up of purely on the protein actually achieves this ultimate mechanical strength is not really known. At the same time, in the case of muscle proteins, so muscle tissue is actually much more complex than spider silk, but yet there are actually some elements which are common between the proteins which actually act as a scaffold in these cases. So in both cases, you can actually treat proteins molecular springs, means that if you know the mechanical property of these molecules, then actually you can model their function. And then main common theme between these two systems is basically the proteins, how actually they are organized in the form of a network. Okay, so the spider silk Okay, again, is made up of mostly proteins at the core, and of course it has some sheath outside skin covering basically to act as an adhesive for basically small insects. But if you see the core, it's mainly made up of mostly one type of amino acid, basically alanine, and the next predominant amino acid is glycine, and other 18 naturally occurring amino acids are actually very, very rare. Yet, actually, this attains ultimate strength because of the complex architecture of this protein. So mainly alanine-based sequences actually organize into what are called nanocrystals, and then glycine-based sequences actually have amorphous structures. So both structured and unstructured regions come together and gives rise to this ultimate mechanical strength. If you see the muscle architecture tissue, it's very, very complex. But if you really zoom in into the cell level or into the individual cells called myocytes, you see very well organized structure of protein materials made up of motor proteins, and where actually they slide against certain filaments. But at the same time, there are molecules which actually connect these motor proteins on the filaments to rigid walls through other protein molecules, and which are actually act as passive elements, which their function is to sense forces and then basically limit the extension of this muscle substructure called sarcomere. But so at the core, basically again protein molecules are involved. Just like in the case of spider silk, where so proteins which are in the structured and unstructured form come together to perform function. So this molecular sensor, which is part of the sarcomere, again has very complex architecture, which is really long polymer chain, longest polymer chain you can really find in the body, made up of structured regions called immunoglobulin regions, where if you see roughly about every 100 amino acids, they have well-defined structure. But however, actually they are interspersed by unstructured regions, which actually don't have any well-defined structures. So basically this architecture is common for all types of tissues. So what do we do in our lab? So to measure the mechanical function of these molecules, either in the spider silk or in the muscle protein titin, so we have to actually work with the individual molecules. So this is where actually I was trying to say this is really throughput. You can really see the one molecule at a time you need to really work with, because there is no way ensemble molecules can be subjected to force simultaneously and then measure its mechanical response. So how do? We actually make meticulously these protein molecules using variety of molecular biology tools, and then subject to single molecule experiments where you can really hold single molecules and then manipulate them using force-based tools and then measure the mechanical response. So why it is important? Because you need to really understand at the molecular level how the force response is actually coming or emerging at the entire molecule or even the entire substructure and entire the tissue cell like myocyte. But what is not really known is that how exactly these molecules, what kind of forces they are subjected to inside the cell. So what is their mechanical response? Can we really understand this in the laboratory setting? So second thing is that, so like I said, this muscle cell itself, the titin molecule itself is so complex that it has many different types of domains which vary actually in sequence. So we need to really understand how the variance in the sequence of these molecules gives rise to a similar structure, yet different mechanical properties. Then we need to really understand the correlation between basically protein structure, and if you introduce any mutations, how exactly it gives rise to basically kind of disease models, because there are mutations in the titin protein which actually cause certain diseases like muscular dystrophy and etc. And this problem is actually multi-scale because you need to really measure the properties at the molecular level, and then you need to really understand the whole muscle cell or the muscle tissue. So it basically scales range from molecular scale like few nanometers to basically few microns, and if you take the tissue, maybe even hundreds of microns. But what are the limitations? This is really the low throughput tool, because you have to really work with molecules. You can't really use ensemble of molecules. And the problem is again difficult, because it's basically applying force to these structures gives rise to different properties depending on how the force is actually transmitted through this molecule. But through rational design and understanding the current understanding of the mechanical properties of certain elements, people actually have started building more complex architectures where you can take protein molecules and then add on components where binding and the force response you can bring into, and then make materials with resilient structures. And by varying the components in the solution, you can actually change the mechanical strength of these molecules. So we want to really make these protein-based self-repair materials, because these materials, because they are protein-based. So for example, let's say if you make a crack here, you can actually repair through protein-protein interactions. And if we make actually proteins with tailor-made mechanical properties, they might be even applicable in biomedical applications. So I'm not really a ML or a DL person, but I would think that this problem requires ML and DL inputs, where we can actually have information which is existing from many different proteins. This is here, the number is just 100. So very small data set, but still we can think that a lot of conditions under which the properties are actually available, measured properties. So maybe we can actually use that data set to even bring in ML and DL to actually understand the problem, and then maybe design based on AlphaFold, maybe new sequences, and then come back and then test their mechanical properties. So with this, thank you, and I'm happy to take if you have any questions. Yeah.
Yes.
Appendages. Slides. One sec. It's the keynote. Here. How come it closed? Huh? It was just open. Can I just share it from Zoom? Yeah. I just want to be able to see the screen anyway. Okay. It was just open. You want to just take it out and I'll. Should I put this mic off? Oh gosh, where was it? It's odd. It's just open. Yeah, sure.
Just again. Yeah. Okay. Yeah, one second.
Why is it not coming up? It was just there.
Stop share the screen also.
It was there. It was just open. I think it goes after you plug this in. Yeah, there you go. Fine.
Open.
Yeah.
No.
Is there? Yeah, fine.
Yeah, it's working.
Okay.
Pointed forward.
Yep. Okay, thank you. Am I audible?
Yeah.
Yes. Okay. So I will not hopefully take too much time here. What I want to do here is to do a little bit of a deep dive into two slivers of our data to illustrate how we think machine learning would help us. I would like to start with these beautiful illustrations from Ramon y Cajal, which are as much art as they heralded an era of neuroscience. So it has been one of the longest quests in neuroscience has been to photograph the brain and to observe structures in minute detail and to understand what changes in a healthy situation versus a disorder. One of the focus areas is and there are several levels of neuroscience, but one of the areas that is under intense investigation is to understand how neurons connect with each other and form these specialized structures called synapses, which are thought to be the business ends of neurons, where electrical information comes from one neuron. On the presynaptic side, it gets converted into chemical information, which is then again detected on the postsynaptic side and converted into electrical information. And this process, the molecules that are involved here, the molecules that keep changing, and the circuits that keep changing, is thought to be the basis of learning and memory. So one of the ways in which in our lab, we look at these structures. So I must mention that these synapses are below the resolution of a typical light microscope. And so when electron microscopy came along, it was a big advancement to be able to observe the fine structure of these synapses and to understand how neurons communicate with each other and send information. In our lab, we developed tools and rigs to look at these synapses below the diffraction limit of a light microscope. And just quite simply to illustrate what we do here is to, instead of capturing one snapshot, we capture a few molecules in each imaging frame. You can do a Gaussian fitting to find out the XY coordinates. And then you build a so-called pointillist image from all these coordinates. How this works in real life is shown here, whereby you can have two things. One is, this is a tissue sample that is fixed, whereby there's a laser that comes on, activates a few molecules. And so because these molecules are not moving, we can iterate this over several thousand frames to get structural information. What you can also get is dynamics information, where if these molecules, so this is a live cell membrane, and each individual molecule is able to move around, you can then get a lot of information about their diffusion and dynamics, which is something I'm not going to talk about today. So using these tricks, you can go below the diffraction limit of a light microscope, and what you can see here is these are two proteins, one on the pre and the postsynaptic side, green and the red. It doesn't matter what the names of the proteins are here, but this is just to illustrate that we can look at these structures now finally using a light microscope. And I want to illustrate one ongoing project to understand what our hopes and aspirations are for machine learning. In one of these ongoing projects right now, it's a collaborative project where we are looking at these hair cells. These are specialized neurons, sensory neurons in the cochlea, which detect sound. And so you have fibers here which deflect with sound. They are connected to the auditory nerve fibers, which send information via these specialized synapses called ribbon synapses. These are present in the ear, and they're also present in the retina. And these use pretty much the same components as the synapses in our brain, but they're organized very differently. And so they're organized very differently. They also send information very differently, because when you have a small amount of noise, you have a few vesicles that fuse, that release the chemicals. Whereas if you have loud noise, you may have more vesicles that are fusing. And so they're typically designed to have tonotopic information that is getting transmitted. So our interest has been to model this structure which so far has been looked at only with electron microscopy, and to understand how this structure is built. So I'm going to show you these two cases whereby these are derived. These are these ribbon synapses. One of them is coming from a normal mouse, and the other is what we are seeking to do here is to compare mice which are born congenitally deaf. And these mice have all the structures intact of the ribbon synapse. It's just that these vesicles are not filled with a neurotransmitter chemical, which is why they cannot transmit. They fuse normally, but they have no information that is getting transmitted. So we decided to look at, Gayatri, a student in the lab, has been looking at these synapses. So we typically, what we do is we isolate these structures from the images. And what I'm showing you here are the postsynaptic side, which is the recipient of the information. And we can detect various receptor molecules. What we found, what we find here is that surprisingly several of these molecules seem to be organized to form a donut kind of shape. So Gayatri went about trying to understand how to quantify donuts. And what we found, interestingly, was that there seemed to be, so these are two different plots here. One of them is from mice that are very young, around 15 days after birth, which is when the onset of hearing is thought to happen. And there are mice which are a little older, 24 plus days after birth. What we find is that if we quantify the shape of the donut, there seems to be a transition that seems to be happening with the onset of hearing. In other words, the donut is less pronounced initially and becomes more pronounced as the mice age when they are getting auditory information. Surprisingly, this transformation doesn't seem to happen in these deaf mice, whereby most of the synapses that we seem to get here do not have this donut shape. There are other alterations as well, but I wanted to use this example to, and also other examples here from our data, whereby we can collect information on these on receptors which are on the postsynaptic side, which are detecting the information. And I'm sorry, this is not visible, but this is a Voronoi diagram over here, whereby we are trying to now also find out what is the degree of clustering of these molecules. And this is being done in collaboration with Vishnu, Kabir, as well as Prasad Perlikar at TIFR Hyderabad. What is interesting here is that several of these methods that we find ourselves using are already are being borrowed from astronomy, because it's these are similar kinds of data, similar kinds of clustering algorithms. The scale bar is different. It's in nanometers over here. But what our aspirations here are to, some of the data that I showed you, a lot of it requires manual curation. A student needs to look at these images. They need to crop them out. We need to rotate them in 3D, reorient them such that we can get the XY coordinates, map them onto a grid, and then get further information out of these synapses. So what we hope to do is that we hope that we can train machine learning algorithms to automatically identify these structures from our single molecule microscopy data. We also hope to increase, because this is such a labor intensive and at times subjective to the person who's doing this analysis, we hope to increase our sample size by a factor of at least 10 or more, so that we are actually looking at, instead of looking at a few hundreds, we are looking at few thousands of synapses, so that we can then realistically capture the variability that exists in different brain regions and in different conditions. So this is one. I wanted to move to the other end of the spectrum, where I am showing you a video from our lab. Now, these are two mice. This is this video has been put through a software that came out recently, which was actually developed for flies, but we can track mice and ask how they are behaving. So what we can do is, using this, we can quantify how these mice are coming into contact with each other, how much time they're spending in social contacts with each other. What these algorithms cannot do as yet is to do what we would typically have an undergrad do, which is to watch hours of these videos and discern, are they mating, are they having aggressive interactions, and more nuances of these. So we really hope that with advances in machine learning, we can do a little more automated analysis of these behaviors to be able to then use these to correlate the earlier structures of the synapses that I talked to you about in the brain regions with what is going wrong in the behaviors, let's say in certain neuropsychiatric conditions. So I think I'll end my talk here. And I guess since there are no questions, we can, I'll be happy to discuss later. Sure. Yeah, what I meant is we are not doing any questions. Thanks.
Yes, it's okay.
You can see it now, right?
Yeah. Yeah, it's coming.
That cannot work, no? It's okay. That work? Oh, great. That's okay. Sorry about that earlier. I'm Uma, and it's great to be here and speak after both Maitreyi and Adi. So that's a line from Wizard of Oz, sorry, which I've adapted. But I work on tigers and bats and many other things. And a lot of the tiger work is resulting from discussions with my ex-student, Anubhav Khan. So basically, well, tigers are going to go extinct soon. And basically, they've already gone extinct in several places like South China, in the Caspian region, and Southeast Asia. And one of the questions which we often wonder about, it's a national obsession, is will tigers go extinct in India? Why, where, and can we do anything about it? The primary cause for this, of course, has been the loss of forest cover, urbanization, increase in population density, which has really fragmented habitats for all wild species across the country. And we've done some work. I'm basically a geneticist. So while I work on wildlife, our lens or approach is to look at their genetic variation. And we worked in this population, Ranthambore Tiger Reserve. I'm sorry, the picture quality is not that great. I'm not sure why. And we basically showed that, the tigers here are isolated and inbred. So what does that mean? That individuals here are mating with each other, and it's like a small family. It's about 70 animals, and they're all related to each other, almost like first cousins or sibs, which is a very high relatedness. And from all of our knowledge from biology, we would predict that these animals should show signs of inbreeding depression, could go extinct soon, right? So in order to understand this a bit more, we wanted to try and reconstruct this, which is the wild pedigree. So this is basically T something else, and all these lines are this tiger may have mated with this tiger and so on. But these are all kind of like, at this point, they're just stories, right? Like a guide says this or the manager says this. But what we're trying to do, oops, sorry, I'm now again zooming forward, is to put this pedigree together by following individuals and collecting DNA samples from specific individuals. So once we have that, we can actually test this pedigree. So that what that gives us is a set of relationships, kind of a realistic or a inference of this tree. But we have no idea what that actually means for their phenotypes. Can we say anything about skeletal abnormalities, conditions which may show up as a result of inbreeding and so on? And so we're really lacking. We have a lot of genetic data, right? Because of next generation sequencing, which allows us to sequence tiger genomes. But we really don't know much about what this means in terms of how these tigers look, what may affect them in the future. So these are the kinds of questions we're interested in. And the idea is simple. It's basically that because tigers are a national obsession, there are thousands of pictures of each individual. And a lot of these are from, for example, this individual. Also, this is just to show that these are all the guys who do the work. This is the team in the field. This is one tigress, for example, and this is a photograph by Jayant Sharma, who's part of Toehold. He's like a professional wildlife photographer, and he just shared with me, several of his pictures. So these pictures, I mean, can we use these pictures, these thousands of pictures, which are 2D, to reconstruct some kind of a model of this tigress? Okay, so this is, for example, the kinds of detailed pictures we can have. These are pictures from my students, so they're not as professional. And you can see it's almost like all angles. I'm just going to rip through these of this individual, for example, right? And because they have stripes, we can identify these individuals quite conclusively. So the number of individuals is small, but we have lots and lots of pictures. These are all the professional pictures by Jayant Sharma of the same individual, right? Of basically different angles, different backgrounds, and so on and so forth. These are all from this park, Ranthambore, and this particular individual. Okay, and Jayant Sharma is just one person who does this. If you go to Instagram, which I'm not on, but I got my students to help me with, you'll find that there's many people who have lots and lots of tiger images. Sorry. So this is, for example, just a sample. So in publicly available spaces, social media, there's tons of images which are available, not just ours. And I think that maybe it's possible to collect all of these images, and I don't know, but maybe it would be very interesting if we could try to kind of reconstruct these tigers and then look at initially very simple things like striping pattern, heritability of those patterns. How does that correlate with our pedigree? Can we detect skeletal abnormalities and so on? I know nothing about how to do it, but it's just a wish in a sense. So the other thing which I have been working on for some time is trying to understand zoonotic spillover. And this is just to motivate that this is an important question. India is a hotspot for potential spillover in the future because of several reasons. We already talked about land use, land cover change being one of them. And amongst animals or species from which we expect spillover, the most likely are bats and rodents. They're very speciose. There are many bats, many rodents, And unfortunately, our knowledge about the viruses or the bacteria they harbor is very poor. So again, what we can do is we can go to the field, we can catch these bats, we can catch these rodents, we can get biological material from them, and we can sequence the hell out of what is there, right? So we can get viral genomes. These are unknown viruses, right? It sounds like a big deal, but it's actually not. Most of the viruses are unknown, most bacteria are unknown. But then what? What do we do with that? Again, our ability to understand which of these may spill over just from a, spillover is separate, but which of these will infect, say, human cells, just from a biochemistry perspective, cell biology perspective is poor. So can we actually use ML-based approaches? You heard about predicting protein structure. That itself is so tough, right? But here we're talking about a whole organism. Can we predict these structures and interactions with cells? And then can we also think about like bigger questions of spillover itself, which depends on patterns of interaction, models of interaction between humans and wildlife, and basically the intersection of hazard and risk. The fact that bats carry these viruses is not going to change, that's hazard, but how we interact with that hazard can be minimized to minimizing risk. So I'll stop there. I hope I didn't take too much time. Thank you.
Girish, are you online?
Yeah, I'm here, Shravan. Yeah,
okay.
Just let me know when to start.
Can you stop sharing this?
Yeah, as soon as the sharing stops. Yes, please. You can share now, I think.
Okay, great. Okay, great. I hope everyone can hear me clearly. So I should start. Maybe I should switch on my video as well. Just give me a second. Yeah, there we go. So I should start by apologizing that I'm not there in person. I had to cancel my visit at the last minute due to COVID, still not gone away. But then I should quickly make sure I thank Shravan for this extraordinary initiative. I think it's a really timely thing to have a meeting like this and an initiative like this. And I do hope that this builds into something impactful and mutually beneficial. So my name is Girish Kulkarni. I'm a member of the faculty at the TIFR in Mumbai. My area of research is astrophysics and cosmology. And I have been asked today to talk about the role of computation and machine learning in cosmology. So cosmology is astronomy at the very largest scales that the human mind can comprehend. Okay, we are talking about hundreds of millions to billions of light years. And it turns out astronomy can be done at those very large scales. And about 100 years ago, almost everyone quite reasonably thought that we little humans will never be able to see the whole universe. But it turns out that at those very large scales, you can begin to study the whole universe as one physical system. So you can ask questions about the physical properties of the whole universe as one physics system. And in the last 5 to 7 decades, miraculously, we have learned a lot of things about the whole universe very precisely. So this is encapsulated by this cartoon diagram by an American group, in which the basics are shown. For example, we think that the universe is 13.6 billion years old. It started in an event called the Big Bang. It had a rapid phase of expansion, which created the various particles of which produce normal physics. And then at some point, stars and galaxies formed, which we see around today. Now, what's interesting about cosmological research is the ubiquitous usefulness of computation, because this picture, this cosmological model that we have discovered in the last several decades is very nonlinear. So almost every single question that we ask requires doing numerical calculations. And the video on this slide shows an example from a really large simulation by the group at Carnegie Mellon University in 2015. So you can see this is an artificial universe being created under certain physics hypotheses inside a computer. Where is cosmology today? Cosmology today is an intensely active field of research. And in my opinion, the next decade is going to be focused around these 5 big questions in the field. We want to understand what drives the Big Bang, what drove the Big Bang, what are the mysterious things called dark matter and dark energy, how did first stars and the intergalactic medium evolve, how do exotic objects like supermassive black holes form, and we want to understand how gravitational waves, which have been now discovered, can be used to study cosmology. Now, the reason things are very bright and exciting is because these questions are accompanied by the development of large astronomical facilities which can get data that are useful to answer these questions. So I'm showing 4 on this slide. The SKA observatory in Australia, which will detect radio data, the LSST experiment in South America, the TMT facility in the United States, and LIGO, the gravitational wave telescope, which will soon have an Indian arm. And these 4 experiments are historic, epoch-making facilities coming up very soon. But I'm highlighting these 4 because they are of immediate interest to the Indian community, but the list goes on to cover about 10 or 15 more facilities. So there's a lot of data coming our way, which should help us to answer those 5 questions that I have highlighted as sort of a consensus direction. The workflow of a cosmologist is like this. You use these astronomical facilities to design an experiment, which essentially involves collection of data optimized to rule out given physics hypotheses about the universe. The experiment runs, produces some raw data, which you convert to usable data, usable summary metrics. This will involve a lot of data processing. Then you go to the theory side and you develop models of the same summary metric under certain assumptions about the universe. And finally, you do the inference in which you confront data and model to either rule out or constrain your model. So for example, if you have your favorite new model to explain dark energy, your prediction might be that that model will form certain patterns in the galaxy distribution on the sky. And then you will design an experiment to measure that pattern. And then you will model that pattern using large cosmological simulations, and you will compare those 2 patterns using some statistical inference technique to check if your hypothesis is successful. So that's what cosmology is. Okay. And now to discuss how computing and machine learning plays a role in this game, I will focus on the last 2 stages of this flowchart because that's my area of expertise, the modeling and the inference part. So when we talk about modeling in cosmology, the central task is to solve equations of gravity, fluids, radiation, and magnetism. That's actually just the task. Sometimes these equations come from conventional physics. Sometimes they come from non-conventional new physics models. And the technical, say, an example is shown here in this animation, a very large cosmological simulation that we published a couple of years ago from my group. Now, the traditional way to do these calculations is to use very large numbers of CPUs and GPUs over MPI. Okay, very traditional HPC setup. A unique challenge, though, in cosmology, which I think probably is not shared by other science discussions that are happening today, is dynamic range. Cosmological computations are unique in that they require very large scales and very small scales to be simultaneously present in your calculation of these nonlinear differential equations. And that makes the problem very large. It has to be spread over gigantic supercomputers. But the task doesn't end after simulation. After the simulation, you should also process the simulation data to get those summary metrics that I alluded to, which you have to compare to the data. And so monthly budget in this business can be easily millions of CPU or GPU hours and petabytes of data. That's the modeling side. On the inference side, constructing one model is not enough. You have to construct multiple models spanning very large parameter spaces, sometimes going to hundreds of dimensions. And then you have to figure out a way to traverse these parameter spaces when you do the inference and compare models to data. Currently, this is mostly done using poorly understood interpolators and the traditional MCMC. As Shravan pointed out in his beginning remarks, this is woefully inefficient, but this is sort of the standard of the field at the moment. And the problem is there are ubiquitous parameter degeneracies, which you have to be very mindful about. I think that is another respect in which it differs from industrial applications of machine learning. Okay, so the summary is that both inference and modeling are extremely compute intensive tasks. Now, this is where machine learning has been helping. cosmology got machine learning techniques only about 10 years ago, but there are 3 applications which have revolutionized certain areas of cosmology thanks to machine learning. The first is increasing the dynamic range by by speeding up just traditional computation using emulations and interpolations using machine learning. Okay, so that's just constructing super resolution, high dynamic range calculations. The second is to target the large parameter space problem, and machine learning techniques like CNNs have allowed maximum information extraction and rapid marginalization on nuisance parameters, which you can't ignore while modeling, but you don't want them at the end of the exercise. And finally, machine learning has also helped in the deal with the problem of the diversity of data sets, lots of telescopes, lots of experiments, and the domain adaptation methods seem to be showing great promise there. So this is the current level of machine learning in cosmology. For our industry partners in the audience, I should point out that cosmologists started by just importing what the industry had developed on machine learning, but we have done innovations ourselves. Our first contribution to machine learning was to adapt techniques to cosmological data sets, which are usually not 2D images, for example, of tigers, but spherical 3D data sets. And I've listed some neural network techniques which were developed in cosmology in my first point on this slide. Machine learning was then adopted to cosmological time series. Again, this has been a very fruitful literature, again, native to the cosmological community. And finally, the latest trend has been explainable AI and symbolic regression. This is showing great promise, and a lot of discussion is happening there. So that's the future, in my opinion, of machine learning from the cosmology side. But I should highlight one respect in which machine learning has failed to please cosmologists, and that is the area of uncertainty and bias quantification. Okay, in industry, often point estimates of various parameters is enough, right? You just have to mark a message spam or not spam, for instance, and so on. But in astrophysics and cosmology, you need interval estimates. You need a confidence interval. You probably need to understand degeneracies. And so traditional regression is not of much interest. Okay, and so there are various techniques available. But this has been the main challenge. I just want to flag it there for any discussion. Cosmologists have therefore liked things like approximate Bayesian techniques, which actually have started moving into the industry. I will end by quoting the recent Decadal survey of astronomy that the American community has published. And these are 3 verbatim sentences from their report. This is what is used by the US government to fund research in this area. And you will see that it really highlights the role of machine learning and computation. Cosmologists have started publishing in machine learning journals and conferences. And I think, in my opinion, the 4 concrete points of synergy probably worth discussing in the afternoon are sort of computing resources, algorithmic development, software engineering, and human resource. And I think there have been efforts, industry academy synergies in cosmology in each of these 4 points, which I can discuss later if there is interest. And I should again apologize that I was not there physically. So if someone wants to get in touch with me, I'm more than happy to talk one-on-one. So I'll stop there. Thank you.
Thanks a lot, Girish. Divya, you want to go next? There'll be time to discuss all of this, Girish. I'm sorry again about not.
No problem. Thanks. Sure. Yeah.
I'm hungry already. I think there is one more, right? There's also one more. Okay, sure. Sorry, I have one announcement. We'll break for lunch after this talk. We'll be back by about 1:45 at the latest. And Microsoft, the person from Microsoft has a deadline. So he says, he asked if he could speak first. So he'll speak, then we'll finish with Sriharsh and Prayush. And then we'll go to Praneeth. Here's Praneeth. Praneeth, is that okay? Yeah, and then the other speakers after. Thank you.
Okay. Hi, everyone. I hope you can hear me across the Zoom. Okay. So What you're seeing on the screen is just a bunch of radio images. There's 48 of them, starting from lower frequency at this end, going to higher frequencies. Each frame is half a second. This is in just a little bit higher than the FM radio band, 140 to 170 megahertz. And what I just want you to notice is all that is happening here. This is just 4 minutes of data. Took something like 25,000 images to make this. So there's a whole lot of stuff happening at the radio waves. Even when actually this is a fairly quiet time on the sun, there's one active region which Shravan showed earlier in his talk. But so basically the point to take away from here is that when you look at the sun in radio waves, it's really very dynamic. Okay, and that's what I want to talk about, how to extract information from these millions of images, which we can now make of the sun at radio waves. Okay, this is integrated flux density from the sun, frequency on this axis, time on this axis, half a second time resolution. All these sort of horizontal stria which you see are changes in the emission from the sun. So as opposed to the visible sun, which sort of looks boring, when you're looking at, please don't look at the sun invisible directly. At the radio waves at low radio frequencies, it's extremely dynamic. There's a lot which is happening all the time, right? There are many different kinds of emissions which have very different structures in the frequency and time plane, which is what I'm trying to show here. There are also span very large ranges in the parameter space. So these red boxes are brightness temperatures. How hot would this body be if it is radiating like a black body? And these blue boxes are fractional circular polarizations, right? So it goes from something like 10 to the power 15 to around 10 to the power 4 in terms of brightness temperatures, 11 orders of magnitude, and some 2 orders of magnitude in circular polarization. So what I want you to remember is that this emission is structured over very small or over a large range of temporal and spectral scales. It shows rapid changes in morphology, 9 orders of magnitude in intensity, and 2 orders of magnitude in degree of fractional polarization, right? So very variable. Just one slide primer about interferometry. It's a Fourier imaging technique. Think of a large number of telescopes as organized pairwise. Each pair we call a baseline. Each baseline measures one Fourier component of the field of view the telescope is looking at. The larger is the number of Fourier components you can gather, the better your image reconstruction is going to be. And I hope I've just convinced you that the emission we are trying to reconstruct is very variable in both time and frequency. So you need to gather your data very quickly in time and over very small spectral spans, right? And that is what we refer to as, and also of course, light is polarized. So if you want full information out of what you're gathering, then you better do a full polar imaging of the sky. So we need what we call now full polar snapshot spectroscopic imaging, right? And that has become possible only comparatively recently. These figures show you the locations in the Fourier plane where this particular telescope is gathering data. And you can see that it's practically quite full, and it allows you a very good reconstruction as opposed to the instruments which have gone before. Okay. These telescopes produce humongous amounts of data, so it's already a challenge to make good images out of these. That's something we've been working on for a while now. Here are a couple of examples from the work by my group. This particular image is the highest dynamic range. By dynamic range, I mean the ratio of the brightest to the faintest believable feature in the image is about 10 to the power 5, best which has ever been achieved. This is about 10 to the power 3. This is when the sun is a sort of amorphous large blob, which is when it is hardest to image. Okay? This particular image is a bit like seeing stars during daytime. This red spot is where the sun was supposed to be, and it has been subtracted out. And there are some 80 other background sources which you can see here. So we are getting to a place where we can really image the sun very well. Okay. I talked about polarization. Here are 3 different kinds of active solar emissions, which vastly or at least substantially different degrees of fractional polarization. So what I want you to remember here is that we can now make images which, with the dynamic range of which exceeds that of the earlier state of the art by at least 2 orders of magnitude, right? And this is naturally enabling explorations of phase space which had never been accessible to us. And whenever you take such a large leap in what you're doing, you're bound to find new things, right? It has never happened in the sun. The sun has never failed to surprise us with a large number of new things which we had not anticipated whenever we have taken such a large leap in our ability to image the sun. So the same has held true for us. I don't want to go through this in any detail, but there's a bunch of new discoveries which our work has enabled. One which I find particularly interesting is what I refer to as winks. These are weak, impulsive, narrowband Quaternion emissions. These, more work needs to be done, but if things go the way they seem to be going right now, they will have a big implication on explaining what is known as the coronal heating problem. How come there is a 2 million Kelvin corona sitting on top of a 5,800 Kelvin photosphere? We find pulsations of time scales from 3 to 30 seconds, practically everywhere we look. For the first time, we have detected linearly polarized emission from active solar emissions, which sort of goes against the conventional wisdom. So it's very interesting to figure out why that is happening. We have detected circularly polarized emission from the quiet sun, which was predicted in 1950s, but nobody ever had the capability to do it because our imaging was not good enough. And that is what we have achieved. And here's an example of that linearly polarized emission. Okay, so where do the challenges lie for us? The challenge one is to go from the raw data which your telescope gathers to build the image. That is what we have been working on. And that there are sort of two aspects to that. There is the algorithmic complexity which is involved. That there is more work to be done, but I think we have reached a very good place. We are the state of the art at the moment. There's of course also the computational capacity. It's computationally very intensive to make these radio images. But right now, it is not our lack of computational capacity which is limiting us. What is limiting us really is our ability to extract the information from these images. Even with our existing telescope, we can make 11 million images an hour. Okay? And I have 3 petabytes of data in the archive. I've looked at less than 3% of that data because even with that, I'm already struggling with my group of students to figure out what these tell us. What is the structure of our data like? Let's say this is the sun. This is say 1 pixel on the sun or 1 resolution element. For each of these resolution elements, I have information about the 4 Stokes parameters, which tell you about the polarization of the emission from the sun. And for each of those parameters, things vary as a function of time and frequency. So right, this is my 5 dimensions in which I have to play 2 dimensions on the sun, time, frequency, and the 4 Stokes parameters. This is just another visualization, which is just a stack of images. The vertical axis is the frequency of how things change as a function of frequency. These oscillations you see, they are real. These are really high quality, high signal to noise data. These structures which you see, this is actually a loop, a magnetic loop on the sun. Okay, so there's a lot of interesting information which is buried inside these images. So the challenge which we are struggling with in our group is going from images to science. How do you extract information from these image hypercubes, right? The 5 dimensions I just mentioned. And like I try to say, all sorts of creatures live in this zoo, right? We have a wide variety of time and frequency structures, large spans in intrinsic brightness and polarization properties. Things move on the image plane, they come and go. There are all sorts of variations. There are physical reasons why some things would grow linearly, while others would have quasi-periodicities. Different aspects have various relationships between them. So there are anti-correlations, for example, in some cases between the area of a source and its intensity. Something which Shravan started or mentioned in his talk, there are, of course, relationships with observations at other wavebands, right? The sun is one large connected system. We are looking at, say, heights of about 1.4 solar radii above the sun. Most of the information which we gather from other sources is much lower down in the solar atmosphere, photosphere, chromosphere. They're all related, right? Things are flowing from down up and sometimes from above below as well. So we also need to look for relationships with what is happening in other observations. And of course, as we go looking for things, you will only find some fraction of what you look for. And you'll, of course, only choose to look for either what you expect or you can imagine, right? So there's always this possibility of missing discoveries just because we didn't think of them, especially when you have taken such a big leap in what you're able to do now. And something like this, a problem like this is, I believe, at least for my brain, it's sort of ill-suited. And also we have more capable telescopes on the way, which will give us even more data, right? So more to drown in. On the other hand, I think it's something which is extremely well suited for AI, ML sort of applications. It's about looking for structures or patterns or relationships in a large multidimensional data set. There are aspects of which can be posed as a classification problem. There are other aspects which should be sort of unguided discovery. I mean, I don't know what is the formal phrase for that. We've been taking some baby steps ourselves, though we've been sort of limiting ourselves only to 3 dimensions in whatever we have done so far. Part of this work has been done in collaboration with E4R, the Engineering for Research group at ThoughtWorks Pune. So that's what I wanted to say about my sort of work. I also wanted to put in one slide for something which a colleague at NCRA works on. This is Tirthankar Roy Chaudhary, who's also a cosmologist, and thanks to the earlier speaker who set up the stage for cosmology. So Tirth's interest lies in studies of what is called the epoch of reionization, the cosmic phase when the hydrogen was ionized by the first stars which came about. And he works in a sort of semi-analytical framework, as opposed to a purely numerical framework, which Girish was earlier talking about. And he uses observations from different wavebands from 21 centimeters in the radio to cosmic microwave background in the microwave part of the spectrum, and also near UV and near IR, optical, and UV to gather the data. The interpretation naturally requires a modeling. Modeling requires simulations. The typical simulation run would take days, actually. But because of the uncertainties in the physics, there are many parameters in your models which you need to adjust to various values, and that creates a rather large parameter space to explore, requiring you to do thousands or tens of thousands of these simulations. And that is where he thinks that the role of artificial intelligence might be useful by trying to have some AI-based emulators to train and predict. And the sorts of techniques he's thinking about are listed here. And what he has just started to work on is on optimizing these algorithms to a particular simulation technique. Okay? And I would encourage you to get in touch with him if you're looking for more information on that. And with that, I'll stop. Yeah. Thanks. Yeah.
Okay, so let's walk over to lunch. Everyone must be very hungry. And we'll reassemble here at 1:45. Thanks.
Yeah, can we leave our stuff here?